now. And we'll have a record of both the questions and this session. It's been invaluable for people on at DivSeek who've come to the website and can see some of the past seminars. Definitely check back and see what the next seminars coming up are. We change that website every week. So from this point on, I'll hand it over to Niels, who can give a little introduction to the scope of the 6 talks.
Okay, thanks, Chris. I share my screen for the brief introduction. I hope at least. You can see my screen in presentation mode? Good. So yeah, welcome everybody. It's my pleasure to be here with you. I saw also our project officer, Verle Lammens, which I really welcome very kindly. I think it's great that you could make it to join the session here. You see the agenda for today. We have 6 talks. Generous Bridge presented by Michele Bolzano and Sheila Kell, For Genius by Ivan Scotti, Agent by myself, Harnesstom by Antonio Granell, Breeding Value by Bruno Messetti, and Gene for Olive by Conchi Munoz Diaz and Christopher Miho. You will ask yourself, why these 6 talks? And this is what I want to introduce you to. So these 6 projects are all funded under the same framework program. So Horizon 2020, it's a EU framework program for research and innovation for a given period. This framework program was put together by the EU by communication with the stakeholders in the different member states and sets priorities, themes, and challenges for funding. The whole program has a volume of about 79 billion euro. And one of the sections is societal challenges with a focus area of food security, sustainable agriculture, and forestry, marine and inland water research, and bioeconomy. And in this focus area, there is a call published called the Sustainable Food Security, with a subcall SFS 28 for 3 subsequent call years on genetic resources and pre-breeding communities. These 3 subcalls were published in subsequent years, 2018, 19, and 20, and we will have projects for all these subcalls presenting in our today's seminar. So it is a unique opportunity that we have a number of projects all funded under the same call for different activities, reporting on either the plans or already on the progress of the individual research. So going back to our original agenda, we have 1 project for subcall A with the theme Joining Forces for Genetic Resources and Biodiversity Management. That's Generous Bridge. Then we have 2 projects funded in the subcall B, Adding Value to Plant Genetic Resources. So the first project, the first call is more on coordination and strategic actions, whereas the subcall B is for research and innovation actions. Subcall C, which will give us 3 presentations, Harnesstom, Breeding Value, and Gene for Olive. These are innovation activities that are already more to the applied area. And with this, I'd like to hand over to our first presenter, which is Generous Bridge. I think Michele and
yes.
Sheila, you will have to share your screen now.
Yes, thank you very much. Thank you for this introduction. As you say, Michele Bolzano, I am the coordinator of the Generous Bridge project. And this Generous Bridge project is a coordination support action. In order to better present this initiative, this way countries have been collaborating, I think I need to provide you a little bit of background. So in Europe, since more than 25 years, 3 networking programs have been operating on genetic resources in Europe. 1 on animal genetic resources, 1 on plant or crop genetic resources, and 1 on forest genetic resources. 3 of them, we were working in parallel, let's say. We're having very limited interactions, but 3 of them we've been working on the same very strong government-based approach, meaning that each country that participates to this networking program, to any of those, basically nominate a national representative. This means that when we arrive to develop a strategy, we arrive to develop a position or whatever, it's not a position of a group of people that happen to be together for a given initiative, but are representatives of all the European countries, meaning that when we identify a need, this need represents the need of Europe as a whole. One of the first needs that we managed to identify as a joint collaboration was exactly the need to join forces in order to learn from each other community and also to develop something in common. So we presented this need to the European Commission that was ready, prone, and proactive in understanding this need, have been publishing the call, the subtopic A on these joint forces for genetic resource conservation. Through this call, these 3 networking programs that actually, well, have a limited number of partners, 17 partners, but through these networking programs, we have about 50 countries that represent a different level that contribute to our activities. So we have been learning from each other, but most important, we have been developing a common strategy for genetic resources in Europe. And this common strategy is basically the main product of this network. And to allow you to follow the process and basically what we've been developing, I will now leave the floor to Sheila Kell from University of Birmingham, who is going to give a presentation about this European genetic resource strategy. Thank you very much. Sheila, the floor is yours.
Thank you very much, Michela. I will just share some slides with you. Okay, so hello everybody, and I'm very happy to present to you today the European Genetic Resources Strategy. I should say that the University of Birmingham was a task leader, a partner in the Genres Bridge project, and we were leading the task on the development of the European Genetic Resources Strategy, and I did this work along with my colleague Nigel Maxted. So I'm going to talk to you about why we need the Genetic Resources Strategy, what's in it, how it was developed, and the current status and the next steps. If I can get the slides moving, good, okay. So just to make sure we're all on the same page, first of all, when we talk about genetic resources, we're referring to wild and domesticated species of plants, animals, forest trees, fungi, invertebrates, and microorganisms, and the genetic diversity within them. And that includes cultivars, breeds, populations, individuals, and genes. And these resources, as we know, are vital for securing options to respond and adapt to the impacts of climate change, for supporting diversification and innovation and the transition to fully sustainable and resilient agriculture and forestry, for improving the productivity and quality of our food and for ensuring nutrition and health, for meeting consumer demands for quality, diverse, and ethical products, and for sustaining the bioeconomy and Europe's competitiveness in the global market. However, genetic resources are threatened by a range of environmental and social factors, are suffering genetic erosion, and are at risk of extinction. And despite their value, they remain of low visibility at the European policy level, and the legal framework, infrastructure, and funding for their conservation and sustainable use is currently insufficient. This is also in the context of Europe's commitment to implementing a number of global policies and legislative instruments relevant for genetic resources, notably the Global Plans of Action for Plant, Animal, and Forest Genetic Resources of the FAO Commission on Genetic Resources for Food and Agriculture, the International Treaty on Plant Genetic Resources for Food and Agriculture, Resolution S2 of the Forest Europe process, as well as specific targets and goals of the Convention on Biological Diversity and the Sustainable Development Goals related to conservation and sustainable use of genetic resources. Sorry, there's some background noise coming from somewhere. So in terms of the current policy landscape in Europe, there are various policy areas that are dependent on genetic resources and/or have an impact on their conservation and sustainable use, with agriculture, environment, and health and food safety being the most relevant ones. But a lack of coherence between them is resulting in a gap in which genetic resources are not being adequately addressed. So to address this gap, the GenRes Bridge project has developed the European Genetic Resources Strategy, an overarching policy framework for conservation and sustainable use of plant, animal, and forest genetic resources in Europe. It complements the EU biodiversity strategy for 2030 and the farm to fork strategy under the European Green Deal, as well as the climate action policies, the common agricultural policy, and the new EU forest strategy for 2030. And in parallel to the development of the overarching strategy, complementary domain-specific strategies for plant, animal, and forest genetic resources are under development, which will address more concrete aspects that are particular to each domain. And these will be published by the three aforementioned European genetic resources networks. That's the European Cooperative Programme for Plant Genetic Resources, the European Regional Focal Point for Animal Genetic Resources, and the European Forest Genetic Resources Programme. And critically, these strategies are needed to strengthen Europe's response to the commitments under the aforementioned relevant global policy frameworks and legislative instruments, and to meet the requirement for greater resilience in agriculture and forestry, for adaptation to climate change, for health, food and nutrition security, and for a strong bioeconomy. In terms of the structure, format and style of the European Genetic Resources Strategy, we took the lead from the EU Biodiversity and Farm to Fork strategies, which were published in May 2020. And similar to these two strategies, it comprises two parts: a short, concise core document of 5 sections or chapters, and an annex containing an action plan. So chapter 1 presents the context and rationale for the strategy. Chapter 2 addresses the priority areas to strengthen and widen actions for genetic resources conservation and sustainable use, and comprises 5 subsections. And chapter 3 addresses the enabling environment required to achieve the actions identified in section 2. And again, this chapter comprises 5 subsections. Section 4 emphasizes the important collaborative role that Europe can play in the global challenge of conserving genetic resources and optimizing their use, stressing the interdependency of countries and regions on genetic resources. We also emphasize here that although the strategy is pan-European in scope, its successful implementation is foreseen to be contingent on political impetus provided by the EU and on the support of its institutions and bodies. The section also places emphasis on the importance of genetic resources to meet the Sustainable Development Goals and of the fundamental difference that Europe can make in helping to deliver them globally. And in the same vein as the biodiversity and farm to fork strategies, the core document ends with a short concluding section. And finally, as noted earlier, the annex contains the action plan, defining recommendations, associated actions, indicators, indicative timescales, and indicative levels of action. That's either at national and/or European levels. The 29 recommendations are associated with chapters 2, 3, and 4 of the strategy and are presented in boxes in the narrative. They define the step changes needed to afford higher priority to genetic resources conservation and sustainable use in Europe. For example, here you can see that 4 recommendations are associated with section 2.2, in which we underscore the imperative to expand the range of species and genetic diversity used in agriculture and forestry, and to achieve this, the need to ensure the availability of and accessibility to genetic resources. And 3 recommendations are associated with section 3.1, in which we highlight the need for coherence, consistency, and compatibility among relevant policies and regulations impacting on the conservation and sustainable use of genetic resources, and for strong and well-coordinated governance between implementing bodies. And if we look in more depth at recommendations 221 and 311, we're now in the action plan. So recommendation 221 is to provide facilitated access to genetic resources under the control of European countries and in the public domain, as well as associated non-confidential data for research, breeding, and training. Then you'll see that there are 2 actions associated with this recommendation. Firstly, to refine and agree guidelines on facilitated access to genetic resources under the control of European countries and in the public domain. That's both genetic resources conserved ex situ and in situ. And secondly, to raise awareness of the international treaty and the Convention on Biological Diversity policy context and practical use of the material transfer agreements for collection holders, genetic resources providers and users. And then you'll see there are two indicators associated with these two actions. Firstly, to the number of countries that have published their guidelines for facilitated access online. And secondly, the numbers of access and benefit sharing training courses provided and trainees with increased ABS awareness. And then the indicative timescales here are within 3 years, and the level of action is both European and national. Then if we look at 3.1.1 to review the existing European policy and legislative landscape and instruments related to or relevant to conservation and sustainable use of genetic resources to identify gaps and needs. So the associated action here is to critically and regularly review existing regional policies. And then the indicator is domain-specific reviews of existing regional policies are published with recommendations for improvements. And again, the indicative timescale is within 3 years for this, and the level of action here is regional at European level. So just to say that in developing the European Genetic Resources Strategy, we've undergone a robust process of stakeholder consultation involving an iterative process of review and feedback on consecutive drafts over some 18 months. Initial input was from subgroups representing each of the 3 domain networks, and this was then expanded to involve the GenRes Bridge project partners and national coordinators of the 3 networks. A major milestone was a stakeholder feedback workshop convened at the end of November 2020, in which some 120 national, regional and global stakeholders were involved. And here the primary focus was to define the draft recommendations. These were then elaborated by an ad hoc task force comprising representatives of each of the networks and the University of Birmingham as task leader. And which also carefully scrutinized the text of the strategy during a series of online meetings to come to an agreement on the precise wording of the narrative to ensure that the perspectives of the 3 domains were properly reflected. A public online survey was also carried out and attracted 76 responses from a wide range of stakeholder groups. And there have also been parallel processes to engage with policymakers, including a study to assess the future uptake of the strategy from the wider policy perspective involving a panel of policymakers. The strategy has been submitted to the European Commission as a deliverable of the GenRes Bridge project, and the document is currently under production in its final layout.
1 minute now, Sheila.
Yes, this is the last few points, thank you. We expect to publish the strategy in its final format in early November, and this will be followed by a webinar to present it in more detail in the first half of November. It's also critically undergoing a process of validation by the national coordinators of the 3 European genetic resources networks, ECPGR, ERFP and U4Gen. And then there will be a launch event in Brussels on the 30th of November with the purpose of publicizing and promoting the strategy, raising awareness of the need to adopt and implement it, and to establish where we go from here in terms of its uptake and implementation. And at this event, the overarching European Genetic Resources Strategy and complementary domain-specific strategies will be presented. Questions will be put to a panel comprising representatives of the European Commission and national representatives of the 3 domains, and this will be followed by a networking event for stakeholders. So that's all for me, and thank you very much. I can't get to the next slide for some reason.
Okay, thank you very much, Sheila. You
There
could.
it is.
Oh, very good. Very good. All right, well, we're obviously going to keep the tempo going here. We're going to save, as I indicated earlier, we're going to save our questions for these individual talks at the end. And so the next talk is from Ivan Scotti in Forgenius. If you could unmute and go ahead and begin to share your screen. Here it is. Excellent. I think you're still muted. There you are.
Yeah, fine. Okay. Thanks, Niels. So I'm going to present the Forgenius project. Forgenius is about finding brilliant ideas about the management of forest genetic resources. And it may not have escaped you that forest trees are long-lived organisms, and they don't move around very easily. And so when they have to face environmental stresses, they have to cope with them on site. And so the first thing that we need to do with forest genetic resources is to understand what happens. In this, you can see here two pictures illustrating dramatic events that have been happening to European forests, wind throws or pest invasions. And so in general, you might say that European forests undergo currently new types of crisis or known types of crisis with increasing strength due to climate change. So this is the first thing we have to assess and evaluate. And the second thing, obviously, to manage forest genetic resources is to understand what to do. And so we need to identify targeted science-based strategies and breeding choices to improve the resilience of forests. Actually, the way we manage forest genetic resources, this is important to stress, is to conserve natural wild forest stands in situ, so that they are able to cope with environmental cues and climate change. So that's a particular way of conserving forest genetic resources that is peculiar to forests. So the project, the acronym stands for Improving Access to Forest Genetic Resources Information and Services for End Users. So there is a stress on the gathering, organizing, and providing information to end users about the properties of forest genetic resources throughout Europe. The detailed goals of the projects are to increase the quantity and quality of information on diversity, both genetic and functional, on the European forest genetic resources, and particularly with a focus on the resilience of those forests. We also need to homogenize the structure of information across countries, species, and plots. Currently, so far, there is a little bit of unevenness in the type and quality of data that are accessible for the different species and from different countries. And one of the goals is really to make these things really homogeneous. And then, once we have done this, we need to convert these pieces of information into tools that allow managers to predict what's going to happen to forests, and so to make decisions about the best strategies for management of forest genetic resources in situ. These things eventually need not to stay in scientists' drawers, but need to be made available through user-friendly tools to the public and to the managers. So the last step of the project is really to make these things user-friendly and available. The project has different work packages as in general happens to European forest projects, and there are 4 groups in general. The upper left group concerns the usage of models and modeling to predict the adaptation or maladaptation of genetic resources to climate change. The lower left half concerns the production of indices about the qualities, properties, and resilience of forest genetic resources. And then the right half of the group of work packages is more about designing services for end users of forest genetic resources information. And of course, very importantly, we need to have a dialogue with the community of end users prior and throughout the duration of the project. I'm going to get into a little bit more details about what we do in different work packages to show you the multi-layered and really multidimensional approach we have in this project. In particular, we're giving nicknames and logos to the different work packages. The first one is about satellites and models. So this work package does modeling and uses heavily remote sensing data from satellite detection and measurements. The second one is called Thirsty Trees, and it's about measurements in the field of the properties of trees that are related to their capacity to stand drought and heat waves. The third one is really resting heavily on evolutionary models. So here we are in more in population genetics, and we relate the future of resilience of forests to their evolutionary potential. Work package 4 collects everything that has been done in WP1 through WP3 and creates synthetic indices of the properties of forests. And then in WP5, we restructure the currently existing UFGIS database that collects data on European forest genetic resources into a new database that is able to accommodate the huge amount of information produced in the previous work packages. The WP6 is the one that will tackle the issue of creating an interface through which end users will be able to access the information stored in the database. And WP7 is about the recurrent dialogue with the larger community beyond science and through in forest management and the larger public. Just an overview of what we use in this project. These are each red dot represents one of these sites where we conserve genetic resources for 100 plus tree species. These are called genetic conservation units, GCUs, and these are run by the UFRGen International Program, of which we have heard before. Just a quick description of the way we use the data. Actually, 100% of these conservation units, so 3,000 and something, will be characterized by remote sensing, and that constitutes the large base of the data pyramid. And this is something that will allow us to characterize the whole conservation network at once. And then we will use genomics to characterize a restricted group of the species and sites, and a further subset of those will be characterized also by the physiological and functional properties. And actually, it is the modeling that will allow us to relate each of the blocks to each other and have a general view of what happens, and will allow us to provide guidelines on how to manage these forest genetic resources. Just to conclude and staying within the time allocated, I'm just going to show you the wise and really multinational partnership that is behind this project, that comprises both academic partners and public forest management bodies, as well as an industrial partner and an international research and policy support organism. And so with this, I wish to thank you for your attention. I'm going to show you the website and the Twitter account of the project. Thank you very much.
Thank you, Ivan. You're extremely punctual and organized, and that was a very clear and effective presentation. Thank you. Again, everyone will be waiting for the end for questions, so write them down and make sure you have them when we open up the floor to general discussion. Okay, so now we're ready for Niels. Go ahead and share your screen, unmute.
All right. Yeah, thank you very much. I'm presenting now the project AGENT. AGENT stands for Activated Gene Bank Network. So you know that there are databases out there which allow you to survey or look for what are the genetic resources that are available for individual crops. I show you here one example of the Genesis website, and I have queried here for the term Hordeum vulgare, and you see that in international gene banks there may be over 250,000 accessions. You can see here also the list of accessions that are hosted in individual national gene banks. Here for Germany, for instance, 22,000 barley accessions, but often you do not really find a lot more information. So how can you really decide what genetic resource is really the right one for your purpose? So how can you make genetic resources more attractive to users? That is part of the focus of the AGENT project by collaboration of European gene banks. So this is one example. You see here glass jars with barley seeds from the IPK gene bank. We have over 20,000 accessions. And if you look at the jars, you can hardly tell a difference. So can you tell the difference? No. So you need to generate and accumulate more information. There's passport information, of course, where accessions were collected. So you may be able to connect this to climate information, but you have often not a lot more information. So we have done a pilot study at IPK a while ago when we genotyped all 20,000 accessions of the barley gene bank, of the IPK gene bank. And you see here a principal component analysis of all these 20,000 accessions based on the sequence diversity information we could collect from these accessions. And you see that the diversity space is structured according to geographic origin of these genetic resources. So having a dense genotype for every accession of your crop species in your collection provides you with new tools to really generate more information about the material. So for instance, if you go back now to the question, can you tell the difference? One important question for a gene bank could be, how unique are your accessions? How many duplications you have? On the basis of this genotype information, we could estimate that about 30% of the accessions are representing duplicates within IPK, based on an identity by state value of 0.05% pairwise homozygous differences. We could also use the genotype information to link it to so-called characterization and evaluation, C&E data, that is usually collected at gene banks during the multiplication cycles. And the group of Jochen Reif here at the Institute has worked on this to see, can we use this characterization evaluation data connected to genotype information and draw conclusions out of it. So what you see in this panel down here is just all the barley accessions in the collection, the year of multiplication. So whenever an accession has a green symbol here in the line, it means that an individual accession was grown out in the field, and agronomic characters like plant height, flowering time, and so on were collected. So you can then integrate this information and look for associations if you have genotype information that is shown here in this Manhattan plot down in the right part of the slide. This is a Manhattan plot for the association of flowering time based on characterization evaluation data and the genotype information of this collection. So you can see that by generating such dense genotype information for an entire collection, you generate added value that allows then also users to make more educated selections of germplasm. This concept of generating dense genotype information for entire collections and use this to underpin the management, so monitoring what kind of selection or drift or multiplication mix-ups may happen during the management, or connecting dense genotype information with additional data generated through systematic phenotyping and precision collections or core collections. We call this gene bank genomics, and it really bridges the gap between the conservation task of a gene bank and plant breeding and research. So AGENT will take this concept of gene bank genomics to the level of a gene bank network. AGENT is a project bringing together 19 partners in 17 countries. You see the blue dots here on the map. We have 14 gene banks involved, 5 bioinformatics centers, because the project is bioinformatics heavy. We are funded with €7 million over a duration of 5 years. We are in the first year, or we have finish the first year, we're in the second year of funding. You see here in this icon the organization of the project. We are having a bioinformatics core that organizes around Eurisco as the main database output that we want to use for the data generated in the project. We have the network of gene banks that are generating new data on their collection, but also activate C&E data that is in the archives. And we connect to an outer layer of additional stakeholders, cooperative programs, and evaluation networks that will evaluate genetic resources that are characterized, also genotyped within the project. And this structure of the project will reach out to public society consumers, the breeding community, policymakers, and research community. So one important concept of the project is that we want to establish a real actively collaborating network of gene banks. And for this, we asked the question, are there mega environments that could help us to organize future organized phenotyping of collections and then integrating data? So we grow general checks. These are 50 spring and 50 winter commercial varieties of wheat and barley each at all partner gene banks. And this allows us to collect agronomic traits for these checks and see how different are the environments in the areas of the participating gene banks. And then we are identifying so-called bridging collections, which are 150 up to 150 barley or 150 wheat accessions of the partner collections, which are potential duplicates between the gene banks. So based on the passport information, we ask, have these accessions been exchanged between the gene banks, which often happened over the past? And if characterization and evaluation data was generated for these accessions, we have kind of replicates of the same accessions from different regions of Europe, which we can then use hopefully for the purpose of prediction or integrating the data sets. So the question is, are there these mega environments and can we use them in the future to design characterization evaluation experiments for genomic prediction in the agent network within the project and beyond? Furthermore, we are working on precision collections. These are 500 barley and 500 wheat accessions from the individual partner gene banks, which have a high potential of uniqueness. So they may represent really unique genetic resources hosted only in this gene bank. So all the collections I introduced you to will be genotyped and we will generate phenotype information. So here for the precision collections, we will collect agronomic traits, flowering time, 1,000 kernel weight, plant height. And this is done at the individual gene banks for their precision collection. We also collect biotic stress information. Here you see this, some gene banks collect information on leaf rust, yellow rust, septoria, or powdery mildew. And this will take place only at individual sites. And then we will have a selected set of the precision collection genotypes from multiple gene banks in a combined collection, which will be tested at a few sites indicated here with the water droplets for abiotic stress, water stress. So the project aims to build an actively cooperating gene bank network that works on agreed standards and protocols. We will activate passively stored genetic resource information from the, in fact, from cardboard. And we accumulate new genetic resource data, phenotypic information, and dense genotypic data. This allows us to, because we don't have the money to genotype the wheat and barley collections of all the gene banks here in Europe, but we really focus on these collections as a concept study to then provide also decision-making, a basis for decision-making at the individual national state level. So we assess the quality and redundancy of existing generous collections. We establish a roadmap towards integrating a global wheat and barley genetic resource biodiversity atlas by generating genotypes for 8,000 wheat and 6,400 barley accessions, which can then be integrated with existing data sets for wheat and barley international collections. There are already large collections genotyped. And we are building coordinated genetic resource training populations for specific traits. At the pan-European level, we want to establish genome-wide prediction across genetic resource collections and establish a coordinated evaluation network of stakeholders. One of the important tasks is also that we implement new ways of quality checking and quality management at the gene bank level. specifically with molecular marker tools to really track accessions and to really track the identity. We work on the basis of the FAIR principles. So we generate data that is findable, accessible, interoperable, and reusable. And we aim to establish new database and novel data mining tools, which will be linked into EURISCO as much as EURISCO will allow, or if not, we have to provide these tools in accessory sites. And we want to work as universal as possible, that it's possible to use the solutions we have provided through the project to translate knowledge into improved genetic resource management also for other crop species. Yeah, with this, just acknowledge the participants here with the icons of the partner institutes and the screenshot from our annual meeting. Like all the projects, we are affected by the COVID situation. These projects, which are meant to really trigger and foster the interaction, the collaboration, are really heavily affected by the non-interaction, because we can't have face-to-face meetings or in-person meetings, which is really a major limitation at the moment, to my opinion. If you need more details on the project and want to follow the progress as it happens, here is our project website. You're welcome to visit and also register for our newsletter, which appears biannually. And with this, thank you very much. I'm looking forward to your questions in the chat.
Thank you very much, Niels. That was excellent. Lots of issues to talk about here later. What we'll do is just keep going through. And so our next presentation will be on harnessing the value of tomato genetic resources for now and the future. Antonio, if you could unmute and share your screen.
Okay, thank you very much for the introduction. My name is Tony Granell. I work in Valencia, Spain. I'm the coordinator of this project, Harnestom, that has to do with not with barley or wheat or grains, not with trees. This is a commodity, a vegetable or fruit that provides you with healthy, nutritious composition that is good for our health. So Harnestom is one of the projects that was funded in the topic SFS 2020. It's about genetic resources and breeding communities, and the main aim is to enhance management and the use of genetic resources and implement global commitment in this area. The project started 1st of October of 2020, so we're barely at the end of the first year, haven't yet done the first year meeting. So why then tomato? Because tomato is the most consumed vegetable, maybe second after potato, but it's the first vegetable that provides good, healthy nutrients to us. So it's very good for our health. It's also important in terms of economic activity. It has been used as a model for fleshy fruit development, but also for other important processes like wound responses or plant pathogen interaction. We have also a large amount of genetic, genomic, and phenotypic information, rather dispersed, although most of it localized in a whole genomic database. And we also have a large amount of genetic resources, but limited. I mean, when I see these hundreds of thousands of accessions that Niels was talking about in wheat and barley, in tomato, we have several thousands, tens of thousands, but most of these are accessions that are tomatoes, Solanum lycopersicum, and with very little genetic diversity. These are basically tomatoes. But when we want to have traits like disease-resistant genes, we have to go to the wild relative, and when we go to wild relative, the genetic resources are rather scarce. So maybe there are a few 100 or more, or a little bit less of some of the related species that are normally what you go to when you want to find these important traits for like disease resistance. So large amount of genetic resources, but limited. It's also a very active community of scientists working that we get together every year. So there's a lot of communication between us, mostly in the academia, but we have to keep in mind that the companies invest like from 5 to 15% of the revenues in research and development. So they are very active in producing new materials to respond to the different challenges. But also in the EU framework, In the previous programs, there have been a lot of tomato genomic projects funded by the EU. And so there is a lot of information, but rather dispersed. Also, when you ask about the genetic resources that there's data banks, they tell you that most of the users are academia groups, but very few genetic resources are requested by companies. So we need to certainly to push this to increase in the use of genetic resources. And the challenges that we face in case of tomato are the same as in many other crops, is the climate change condition, emerging diseases, but also changing consumer needs. Consumers now want better flavored fruits or more nutritious or healthier fruits. But in the project, we also have to take into consideration that the different interests of the farmers and versus the seed companies. Also the different interest of the public versus the private, and also differences between the traditional tomato varieties and the modern varieties. They're normally RF1 hybrids, while the traditional varieties are normally non-hybrids. So what is the approach that we took in Harnestone is we have like two big groups of tasks or work packages. One is to build a centralized one-stop shop for tomato, genetic resources and information and tools. This is very highly emphasized, but also including those tools to increase breeding efficiency. We also have, and this is, let's say, almost every partner participate in this huge task. But we also have built targeted alliances between the academia groups and seed companies and farmers. This isn't the kind of ring-fenced type of pre-breeding project, because we know that companies don't like to share a lot of information with others. In this case, for specific traits, for specific goals, we have this ring-fenced type of pre-breeding projects. We also have another kind of approach that we use for the interaction between academia and farmers. And this is for the case of participative breeding, in which we have farmers, but also chefs and consumers involved in this participative breeding. I will explain this later in more detail. So about the one-stop shop for genetic resources, genomics, and breeding. So we are lucky that in the previous year, we have been most of the partners in Harnestone were also involved in European projects funded by the EU, like Traditom, Bresov, UTP Sol, TomGem, Root to Power, etc. All these were projects aimed to specific stresses or specific sets of variants, like traditional tomato variety, there's adaptation for low nitrogen use, or high temperature, or general diversity in the tomato clade. So the idea in this work package is to collect and centralize the genotyping information and the phenotyping data produced in all these previous EU-funded projects. Also to implement, search, and analyze, and use and develop new tools for visualization, so that different type of users, farmers, breeders, researchers, can use the information in this one-stop shop. And most important, we'll light this information stop shop linked to the germplasm banks. Okay, so most of the germplasm banks are located in different countries, but so far, as Niels also indicated, basically after the name of the accession is the passport data and not much more. So we have over the years with this project, we have generated a huge amount of genotypic and phenotypic information, and further additional one we'll produce in the Harnestone project that need to be centralized and linked to the germplasm banks that are the ones that conserve and the seeds. Okay. An important work package is also about the tools to increase the speed, efficiency, and precision of breeding. Even in tomato, we can make 2 or 3 cycles a year, but it takes several years to get a new variety, because there are a lot of back crosses and to do, even if you do 2 or 3. So in this work package, we have been using, we propose to use a number of different technologies, both genetics and also biotech technologies, transient expression of some factors, to reduce the generation time. Also, similar approaches, genetic and biotech approaches, transient approaches to increase recombination rates. We are lucky that we already have some large population well characterized to analyze the genetic basis of some of the traits, but we still need to do additional genotyping and resequence some of this collection. And this is also a set of tasks in this work package. And we will, although in tomato there are no so large experience in genomic oriented selection to enhance breeding gain, this is also one of the purpose of this work package. And this will, of course, will come to increase the description of a number of materials of population that will help to increase pre-breeding efficiency.
Antonio, a couple of minutes.
Yes, yeah, sure. The other set of work packages have to do with, as I said, ring-fenced type of pre-breeding program in partnership with academia and companies. And we will be addressing the climate change, this emerging of disease resistances, and the quality, fruit quality. And in all the three work packages, the same structure. It's compilation of literature, information databases, and our info. In the case of this new disease resistant, of course, we need to find new source of resistance because these new pathogens just emerged a few years ago, and we're still trying to identify new sources of resistance. But for those other traits like heat tolerance or quality traits, we already have information about markers associated with trait, and we will implement in collaboration with each of the participating company a specific marker resistant plant breeding programs, also pyramiding some of these traits in the same background. And for this, we use both academia lines and elite lines from the company. The idea is then that more advanced materials will be tested in different conditions at the end of the project. In the participatory breeding, we will get the farmers involved, but also consumer influential chefs in defining ideotypes, build a repository of genotypes, and then to work with them in a participatory breeding for improving the efficiency of the breeding, but also to look at the evolution of land races and also to provide new variants in terms of color of fruit quality. So the expected outcomes is that we build a strong alliance, we build a public repository of all this information, we develop new tools and materials to increase the speed and precision of breeding, and we show with some demonstration cases that the genetic resources in tomato can be used to develop new materials that are of interest for breeders and farmers. And how we do this, the most important asset of the Harnestone is the partnership. It's 22 partners, 12 from academia and 6 companies and 4 NGOs from 7 countries. In total, we'll be working close to 1,500 person a month in a project that will last 1,500 days. So basically, every day, 30 people will be working full time for the project with a funding of a little bit over $1 million, 7 million of which will come from EU funding. And this one slide we can leave for the discussion because this was a slide that prepared for the possible interaction and synergy between EU funded projects like we are participating in. So I will leave this for the discussion. Thank you.
Thank you very much. This is brilliant. This is really interesting, very interesting project. We're going to move on to the next sort of keeping the vein with pre-breeding. We're going to hear from Dr. Bruno Mazzetti, the Breeding Value Project, pre-breeding strategies for obtaining new, resilient, and added value berries. If you could unmute.
And please go to presentation mode.
I need to unmute first.
Yeah, and then go ahead and share.
And I go to share. I was a little bit in hurry, and now it should work. You see, no, my presentation.
Excellent.
So, good afternoon. Good morning and good afternoon to everybody. My name is Bruno Mazzetti. I'm the coordinator of Breeding Value, pre-breeding strategy for obtaining new resilient added value berries. It's a project founded by the same program in the last call, as it has been introduced by Antonio. the tomato project. Why berries? Berries generally are considered small fruit, but in reality, the interest on berry it has been increased quite a lot in the last years. And now, because the consumer appreciate berry, we see that there is really a continuous important growth in berry consumption in the world. And the rate of growth is still considered quite high for the next period. Strawberry represent the major berries, but raspberry and blueberry are also considered now of high interest and in continuous increase, and particularly for blueberry in the last few years, the increase has been quite high. Berries are important for Europe, even if we know that United States, China, Russia, for some aspect, are the main producers for these berries. But if we consider the different European countries, we see that Europe, for many of these European countries, berries are quite important. So this probably is the reason why the European Union accepted to support this project. Well, I give you now the key facts of the project. It's again a 7 million project. Started January 21, 48 months, 20 partners from 8 countries. Among the 20 partners, 7 are SME, small medium enterprise. Just to underline the fact that this last call of the GenRes program was specifically addressed to the transfer of the knowledge and the technology on genetic resource also to companies. So we were really working with the idea to discuss and see together how the genetic resource can have an impact on companies. And then we will see also clearly the impact on our major stakeholders that clearly are the consumers. Actually, we also have a tool that are called Target Call, that we allocate money on this tool for further involve additional companies. So we believe we are having now even a call open for having additional companies involved. At the end, we will involve additional 19 companies. Here you have also the link to the webpage of the project and Twitter, so you all are invited to look at this and be in connection with us. Why the breeding value is important? We realized from this paper that was published in 2009 that domestication, like in all the other crops, had a very bad impact on biodiversity and in loss of variety, but in particular loss of allelic diversity in the cultivars that now we have in our cultivation system. So practically in the last less than 50 years, most of the about 35% of this biodiversity was lost, and only in practically 10 years, almost 20% of this allelic diversity was lost. So there is really the need to look at this issue also for berry and avoid to continue this degradation of biodiversity and actually go back, try to identify the biodiversity and work together with the company in order to use it because we need it in order to solve many issues affecting strawberry and berries cultivation in general, considering all the main aspects that we have seen already introduced by our colleagues, so climate change, new pests, new diseases, and so on. So the aim of the added value was to investigate the domestication effect by determining the allelic diversity and answering to the main question. So how still is resilient our breeding base and how this will support our future in future breeding program for this crop. And so we, in the past, there were already different national and EU program working on berry genetic resources, in particular strawberry. But this project is starting from what was already done in the past. And when we were proposing the project, by looking at the different material available or the different information available, we decided to start with this kind of plan of characterizing the different material available in 6 main categories. So the really, let's say, old variety, so until 1960s, that still are available in some repository. Then the modern cultivar, so in the last practically 45 years, sorry, 1960, 2005, and then the newest released cultivar from 2005 to now, so what we have now on the market. But then also, what is the pre-breeding material? So if some of the breeding program are including genetic resources in the breeding program. So identifying pre-breeding materials that it's including the use of genetic resources. So see how far is the variability existing in this pre-breeding material. And then also wild species, because clearly there are wild species available, not many, but still available, and population. So this is a very large amount of material that clearly for strawberry is really much larger. In fact, we identify practically more than 1,500 accessions that have been identified for the major five categories, not including the breeding population, made available from the different partners for the project. Some of the partners that have the repository are providing much more material, but also company were providing some of these genetic resources. So this is clearly a large variability. And you can see the difference between strawberry and blueberry and raspberry. So only a little bit more than 80 accessions were available for these 2 crops. But and practically only few partners, only few institutions are still maintaining a good repository of this material. So it's really important to develop much more the work on these 2 additional crops. So practically this is what we have done in the first 9 months of the project, because I'll say the project started in January. So now we have done this work of discussing with our partner and try to collect all this information, and this is the material that we have identified, and on this material will be developed most of our work in the next period until the end of the project.
1 minute now. One more minute, 2 minutes. Okay.
Yeah. Okay. Yes. So this is the structure of the project. So all this material will be maintained in the field. So different partners will manage the field. On this material, we will apply advanced genomic and genotyping and phenotyping tool, and we will see the stakeholders, the consumer are really important. So we will focus on this, and clearly data management. We will have bioinformatic groups that will help us to manage all these data. But also we included LCA analysis to assess the real improved sustainability of some of this genetic material. So the work here are summarized different type of techniques, genetic resource of what we have done, identify genotyping tools, marker assisted selection, new genomic selection studies will be developed, phenotyping, quality parameter, image analysis, plant architecture, metabolomic studies will be applied, and then the consumer science. I said here, as we have a strong connection with company, we also will try to handle the issue related to sharing the technology, but also the intellectual property management of the new materials that will be generated by this program. The consumer is really the main issue, so we will focus on communicating with experts and consumer, and we will try to better understand what is the real appreciation of the consumer, what the consumer wants for this type of appreciate for this type of berry, depending also to the type of consumer from the south and from the north. So, and for example, the first issue will be the definition of the lexicon, and try to identify people that all together will work in order to have a common lexicon for describing the quality of this berry. Just now we have opened a call for having 7 additional companies involved for this work. In the conclusion, for berries, we expect to improve the information status of the berry genetic resource, to speed up the introduction of usable characteristics for GenRes into berry breeding. So that's really important to improve the efficiency of the breeding. Clearly, for the EU genetic resource program, align berry genetic resource to EU GenRes, contribute the developing of the genotyping and phenotyping tools. This is the interaction that we would also expect to have with the other EU project now running using this, work with the participatory approach, and particularly interacting with company, but interacting with consumers, so have this kind of public and private activity strictly related. And as mentioned, considers the management of the intellectual properties that will come out, will be an issue for the companies that are involved in using this material. And clearly, at the hand, increase consumer awareness on the benefit of using genetic resources for releasing new improved berries and in general new improved plant. I stop here and I thank all partners for helping me in preparing this presentation.
Thank you very much. It's ambitious. It's a very integrated system here. Really nice. So if you could unshare your screen, and then we'll move to the final presentation for today, which I'm really interested in, is Gen for Olive, bringing olive genetic resources closer to breeders and growers. And Dr. Diaz, if you could go ahead and unmute and share your screen. I can hear you.
Yeah, can you hear me?
Yep.
Can you see my screen?
Yes, perfect.
Okay, then full presentation, is it okay?
It's great.
Okay. Good afternoon. I am Conchi Muñoz Díaz from the University of Córdoba. And I would like first to thank DivSeek for giving me the opportunity to present Gen for Olive. Sorry. The olive tree is the most extensively fruit crop cultivated in the world, with more than 11 million hectares. It has greatly surpassed the limits of the Mediterranean basin and is currently in the 5 continents, mainly because its 2 main products, the olive oil and the table olives, which are largely responsible of the health benefits of the Mediterranean diet. Olive genetic resources are composed by the cultivated olive with a wealth legacy of clonal cultivars. Also the wild olive, which is the ancestor of the crop, and is in many areas of the Mediterranean basin with a low probability of frost. And specifically for this species, we also count on the very ancient olive trees, both cultivated and wild. These trees are a cultural legacy, but also are a kind of fossil genetic diversity. The olive genetic the olive genetic resources are facing several threats, most of them leading to genetic erosion. For example, in the crop, this super high density system and spreading worldwide, and they are mainly based on 3 clonal cultivars in 3 clones, which are the only ones that so far show the low vigor that is required for this planting system. The wild olives are facing, the populations of wild olives are facing fragmentation, and in many places are rampant integration from the crop. The ancient olive trees are also suffering these threats, and also are being Many of them are being pulled out and sold as ornamental trees. Overall, climate change is affecting all of them. The Mediterranean basin is a very fragile area, according to all the climate change predictions. Climate change is triggering very devastating disease outbreaks like this one produced by Xylella fastidiosa in the south of Italy, or devastating wildfires that this summer we have seen from Turkey to Spain that had burned to ashes the crop, the wilds, and even very ancient trees like this one, which was supposed to be more than 2,000 years old in the island of Evia in Greece. What can we do? What are we doing to counteract these threats? First, all the conservation actions in olives are based on ex situ conservation. There are more than 90 live cultivar collections around the world. 3 international collections that are recognized by the International Olive Council, but unfortunately, in many of them, the identification is still a pending task. And moreover, the characterization of the agronomical traits of these cultivars. Many of these collections are not collections of cultivars, are collections of accessions, because they are not being correctly identified still. These collections have no representation of wild olives, and to our knowledge, there isn't any seed collection that represent the diversity that still is present in these populations of wild olives. Breeding is key to face climate change, but breeding is still germinal for the olive tree. Less than 20 new cultivars have been released, most of them with very scarce diffusion. And olive breeding face very important methodological issues. For example, it's difficult to cope with the juvenile phase of this species, which in natural condition can last 15 years. Also, the lack of agronomical information about the cultivars is hindering the breeding. Because of that, most of the breeding initiatives around olive had been developed by public institutions. In this scenario, we thought that an international initiative that should promote the evaluation of olive genetic resources, integrating most, or at least the most important present banks, making the information of this evaluation available to all the stakeholders to boost breeding and also to promote an efficient conservation of the genetic resources. In this way, GeneOlive came to life as an European project funded by European Union under the topic genetic resources and breeding communities, with a duration of 4 years and an overall budget around 7 million euros. The coordinator of the project is the University of Cordoba. The project is composed by 17 partners, 5 of them enterprises. The different partners are from 7 countries, and the 3 international banks and 2 complementary banks are also participating in the project. Which are the main goals of Gene4Olive? First, to characterize olive genetic resources. We want to know about their genetic resources, their resilience to climate change, their resistance to pests and diseases, their production and quality traits, and their possible adaptation to modern planting systems like these super high density systems. We want to elaborate common protocol to easy and with reliable conditions perform these evaluations, and also to develop new biotechnological tools that allow us to speed breeding, to efficiently select seedlings in early stages. We want to do an efficient conservation of olive genetic resources, promoting the cooperation among germplasm banks and harmonizing the evaluation and conservation protocols. All for breed the cultivars of the future. We will need cultivars adapted to climate change, resistant to pests and diseases, and suitable for the new olive growing system. And overall, we want to make this this information available to everyone. Sorry, but this, I don't know, this is not, sorry. Okay, we want to make this information available to end users. This, the Gene for Olive project is composed by 11 work packages, but these 7 are the core of the project. In the work package 1, we are, all of them, we could say that are running in parallel. In the work package 1, we are defining and elaborating the common evaluation protocol. This step is almost done. In parallel, the collection of wild olive trees have started in the Mediterranean basin. Also, we are starting the evaluation of olive cultivars in the 5 germplasm banks that are participating in this project. We are evaluating chilling requirements, tolerance to drought, to pests and diseases. Also, the diversity in of the olive oil composition. We want to know, for example, if it's possible to breed special cultivars with a specific composition, phenolic composition in their olive oils. We also are trying to develop new tools to speed up the breeding process to cope with this long juvenile phase, also to try to select genes involved in resistance to pests and diseases. And also we are taking advantage of a group of around 30 cultivars that are present in the 5 germplasm banks to assess the genetic per environment interaction. And also to elaborate models that are able to predict the impact of climate change in cultivars and specific traits. All this information will be available in a database, and also we are building apps to able to recognize cultivars and also pests and diseases through image analysis. Also, for us, it's very important to promote the involvement of enterprises in olive breeding. And because of this reason, this project counts on 2 open calls for enterprises in which we want to enterprises, the private sector, get involved in olive breeding. So currently, one of these calls is open. All in all, what we pretend with Gene for Olive is to characterize a large number of wild and cultivated genotypes under common protocols, promote the efficient conservation of olive genetic resources, make information readily available to breeders and end users, and develop efficient markers and techniques to accelerate breeding, predict the possible or the effect, the real effect of climate change, to try to find solution. We need the private sector to be involved in olive breeding, and in summary, lay the foundations for the future olive growing. And thank you very much for your attention. And okay, this is the webpage of our project in case you want to know more about it. Thank you very much.
Thank you, Dr. Diaz. That was very interesting, very integrative. I guess what we're going to transition now to general discussion. I'm sorry we're not, have the full 25 minutes, but these are pretty complex integrated talks. I think we'll start with one question that's been already posted to the chat, and if you could. Those of you who have particular questions for each of these projects, do refer to the chat area and we can in turn discuss it. Sheila, do you want to ask your question directly, or should I read your question? Or everyone's?
I can ask it if you like, if you want me to.
Yes.
I can remember what it was. Yeah, so Concepcion, thank you very much for your talk. I love your project, and especially the logo, I have to say. Just wondering, you talked about germplasm conservation in gene banks, and I was just wondering whether there was any consideration in this project or any other associated initiatives for in situ conservation of olive, both wild and cultivated.
Okay, in this project, what we want first is to have an idea of the genetic diversity still present in the wild olive. I am completely agree with you that the in situ conservation strategies are the ones that apply best to the wild olives and also the ancient olive trees. But regarding wild olive, we first want to have an overview of the genetic diversity of this population to know which one are the most important or the most valuable to protect. Right now, there is not any specific policy to protect wild olives or ancient olive trees. They are protected if they are in a protected area, if, for example, if they are in a natural park and so on, but there is no any special figure of protection for wild olives. For example, in some areas, the wild olives, the introgression of the cultivars is so rampant that maybe, I don't know until what point is valuable to protect that genetic diversity, you know? But we want to evaluate that within this project.
Thank you.
Thank you very much for the question.
Great. Any other questions? Do post them to the chat. I have a quick question for Ivan. In your presentation, which is fascinating, all the different interlocking components of the Forgenius project, you have a modeling component, clearly, demographic model. Has there been any discussion about sort of assisted migration and essentially reforestation based on predictions of climate change? Because the UBC, the University of British Columbia, has a fairly active program in that area.
Yeah, I know it quite well, indeed. So to say, yes, what we are going to provide as a result in the modeling can be used for assisted migration. The focus here in this project is about characterizing and protecting the existing network of genetic conservation units, which may, of course, require moving some of those germplasm collections somewhere else, in case we realize that they cannot stand the climate change where they are. So yes, this is one of the potential outcomes of the project, for sure. Yes, and of course, the way we see the forest managers using the information is also to go and collect genetic resources that are predicted to be better somewhere else than where they are now, and to replant.
Great, thank you.
Like,
I believe, Nigel, you asked a question of Niels early on. I don't know if it was answered directly, and it was an ecogeographic question, so I'm very interested.
It was, but perhaps could I start by just following up on the question that Sheila made to Conception, that I worked on a project on wild apples in Kazakhstan, and we had exactly the same problem there with cultivated and wild apples, the wild apples being polluted by cultivated material. So what we did there is we set up a field gene bank that was high in the mountains, away from cultivated areas, and we transposed some of the plants that we thought weren't genetically polluted to that location, so they would be preserved, and then there wouldn't be intercrossing. So maybe that's something you need to think of in terms of olives. So the question to Niels was about, basically it sounded as though your project was looking at trying to maximize genetic diversity recognition and using genomic techniques. So the first question was, you said that you couldn't characterize using genomic techniques all of the accessions that you included in your analysis, but you didn't clearly explain, or sorry if I missed it, how you chose the ones you did include. So I would like to suggest, if you haven't done it, to use some ecogeographic land characterization techniques, which are a kind of, rather than just using traditional country boundaries, using genetically identified units using an advanced ecogeographic technique. And then, sorry, if I may have a second question. So the second question was, your project seemed to be assuming that genetic diversity was located in the gene banks. But we all know that the gene banks don't always have a perfect representation of the genetic diversity that's found in the wild. So I wondered, in your project, are you going to look at some on-farm material of land races of wheat which are not represented in the gene bank? Then again, you could use ecogeographic land characterization techniques to identify these particular populations that you'll sample and compare the material held in gene banks with the material outside. So then you have an assessment of how well the gene banks themselves are conserving genetic diversity. Sorry to ask such a long question.
Yeah, it's a long question. I hope I can recall. So the last question about are we planning any in-field collection and so on. So I think for wheat and barley, we are in a relatively clear situation that at least the gene banks involved are not in areas anymore where land races are grown that would provide diversity that is not yet collected in gene banks, as far as I know. There is at least little chance that we would find uncollected diversity in Europe. But
so
we
if I
have.
can exclude for a second, one of the projects I've been working on within the UK is looking at the Scottish Islands, and they have a particular land race of barley there called beer barley.
Yeah, I know.
And although there are some accessions in the gene bank accessions, we think, don't represent all of the diversity that's present on the islands.
Nigel, we could discuss this for a whole evening, I'm sure. The point is that we have not been able to include all the gene banks we would like to involve, because these competitive calls at the EU level lead to the situation that gene banks are involved in competing project proposals. Yeah, there are major gene banks that we would have loved to include in the project, which are at the John Innes Center, which are the Nordic Gene Bank. And so what we know is that based on the study we did at IPK, that at least we have already covered a substantial amount of the global diversity. We have ICARDER in the project, and we have the Vavilov Institute. We use the project to lay out a strategy plan, which then has to be taken further in the individual gene banks by characterizing further material comprehensively. ICARDER has set out on this already individually. Yeah, so the CG centers are systematically characterizing genotypically their collections. They have done it with maize and with wheat at CIMMYT, and ICARDER has done it as well on part of the wheat collection and also the barleys. But I agree, this project will not solve all the problems. It will allow us to highlight certain areas where we can make improvements. The other question about the ecogeographic information is an important aspect. We try to include as much as possible such information as well. You know that ICARDER has implemented the FIG strategy, and we are trying to use that information also for the further data integration as we have collected then the phenotypic and genotypic information to make predictions for the other collections, which are not necessarily fully genotyped. But it's a complex thing, and we can probably only give a flavor in the project, which will leave room for additional research in the future. But you're addressing very important questions.
Great,
I
thank you.
see. Let me take the opportunity, as I see Annette Schneegans, who is the DG Agri Policy Officer, who was involved in setting up quite a bit of these programs. And I see also Welle Lammens is still here. I don't know if we are at the forefront of organizing such joint events of projects funded under the same call. I wonder whether also from the official side, this kind of joint event is seen as a useful activity. I don't know if Annette is still there.
Yes, I'm trying to log in with my camera.
Sorry,
I can see speeding up, no?
Socratic.
No, because the sun was, I mean, if you can see me a bit. So no,
We can
thank
see
you
you
very
and
much.
hear you.
Yeah, okay, great. No, thank you all for your presentations. And actually, it was very exciting to see that the 3 projects and then, well, another one from the same call gathered without, as you say, without us asking you to do so. I think it's extremely useful. We had actually in the topic, we had repeated the request that we work together one way or another. What we do normally is we have a couple of, how do we call them? So events where we put together projects working in similar areas. We have, for example, tomorrow, a cluster of projects all on plant health. These are, however, major, well, bigger events where we have, let's say, projects across various topics, then with close involvement of the REA, of the research agency, that is your project manager, so to say. So it's more formal, okay? And we have this kind of cluster events. And I think it would be a fantastic idea to have something similar for the ones that we label under genetic resources. And in particular, the ones where we have the gene banks involved. We have some that even just finished, but still would be good to capitalize on their results. And yeah, so I would really like to take this up on board. And in particular, in your presentation, Niels, I was very happy to see that you are looking into harmonizing some of the protocols, quality management. I had thought that this had happened already in previous actions. So I would be very keen to learn to which extent that happened, and yeah, let's say, where are the bottlenecks if this didn't happen? Where are the problems? This is all something that I think you have all a much better view, but this is something that we had in mind already with the topics in 15-16. And so I would appreciate very much having a very open round discussion to capitalize on existing results, but also to lay out what is still to do. And as you said, you will not finish yourself neither. You set an example for barley and wheat. How can we use this for other crops and continue?
Yeah, thank you. I think that would really make sense to follow up on this in a broader level. But there is more work to be done.
Yeah, I think the quality management aspects, the data integration that we see across all of these projects that connect characterization and evaluation data directly back to the gene bank and enable them to make better management decisions is really an ongoing effort here, and it's being led by major projects, and hopefully we can establish those best practices and apply them more widely to other gene bank operations. I think that's, well, it's one of the objectives of DivSeek, is actually to try to encourage and enable that networking, to try to take examples from good projects, whether it's tomato or olive or barley or strawberry, and apply it more widely to undersourced collections as well.
Since I have all of you all here with your expertise, would it make sense to have, I mean, these are all research innovation actions, the ones you presented here, but would it make sense to have a dedicated topic, a coordination support action, one that is not under pressure to really big research and be excellent, when in evaluation terms, but really take stock of maybe starting with some crops, but take stock of all the issues of gene banks and what the bottlenecks are and what the results of the projects was. Would that make sense, or is it just would it be just delaying a bit the actions? You see, if we program now a CSA, coordinated support action, it would probably not start before, let's say, 1 year, result in 3 years. So would that make sense, or rather not? What is your feeling?
I think that will always make sense, but there were already similar activities. Maybe Theo van Hintom would be willing to
I was going to say,
answer to
me
that
too.
question. But my personal view, Annette, is even there were already similar activities perhaps in the past. Having, again, the opportunities to bring together the community is important, because we are working really with different different speed at different places in the world, and it is important to really interact with the different stakeholders in that area again and again, and revisit what advances were made also in research projects in other networking activities. As you could see already from the presentation of these 6 projects, they are all tied together at the common theme of genetic resources, but they were very diverse, and it is not necessarily straightforward for us as project leaders to identify really the individual aspects which will allow us to go really into deep networking and taking advantage of synergies between the projects. So I think if there is more of the opportunities of bringing together the gene bank and genetic resources, I include here also the botanical gardens, as I see John Dickey here, is really an advantage for the community. But Theo, if you're still there, maybe you have an opinion to share with us.
I am still here, and lots of opinions to share. But I think that Niels, with your remarks, you already cover a lot of the territory. Perhaps, and we've been discussing that in ECPGR context a little over the last few weeks. It is important to recognize that listed in the FAO databases, we talk about thousands or hundreds of gene banks and millions of accessions covered with those gene banks. Perhaps we should be a bit more strict. Perhaps we should get a bit more clearer what a gene bank is and what minimal requirements should be set for a gene bank to qualify as a true gene bank. Having to do with the conservation component, having to do with the access component, the standard operating procedures and so forth. And I think we're at the stage that we could define them and set up a program, at least in Europe, that would distinguish the true functioning gene banks that can rely on each other. Because if somebody else does it, we don't have to do it, right? But that requires a certain level of quality. So we have to set up a quality assurance system. We have to make clear what standards need to be applied to meet the criteria that we think are necessary. In parallel, we should have activities making sure that gene banks that don't meet those requirements yet can learn to reach those requirements. Once we have that a bit more defined, we can tackle issues such as how can information best be managed? What are the common problems in the true functioning gene banks regarding the use of the molecular data that is being generated large scale, and DivSeek should play an important role in that, right? But at the moment, we're talking about such a wide variety of actors and different objectives and serving certain communities, but not others. Most gene banks have much too much material, so they can't cope with the regeneration and proper management of the stuff in terms of germination testing and regeneration, etc., etc. There is an issue there. So I would strongly support having a wider discussion on these topics, involving also CG, for example, and the Global Crop Diversity Trust with their way of approaching the gene banks can learn a lot to the rest of the gene bank community in the world, certainly also in Europe. On this topic. So thinking about setting up a platform for having this type of discussions, what to do to be a real gene bank, and how can we help each other to get as many gene banks involved in that network, that would be great. That would be great. Whether we're ready for that on a global scale, I'm not sure. I think in Europe we could certainly do that. And within Genna's Bridge, ideas along those lines have also been expressed. So the material is there, we could do it.
Can I come in and just add a point to what Theo's wisely already said? I agree with everything he said, but I'd just like to please beg that we don't forget on-farm and in-situ conservation when we're doing this review, because I think we're also at a stage now where we can move seriously ahead with on-farm and in-situ conservation, and move from talking about it to actually doing it. And there the gene banks really are a million miles ahead of us, because they've been doing it for a very long time already. But we're at the stage now where we can actually apply these things. So I really think we need to include that in this discussion as well.
I may say, in addition, I fully agree that all this is a really important issue. But as I was also already trying to say in my presentation, is how to manage the transfer of the knowledge and of the material to the industry or to the company, let's say, to the production system that want to take advantage of this genetic material. So I think that is also an important issue to be managed. And at the end, it's also the issue that bring to the final use and valorization of the genetic resources. We are discussing this with the company, but you know, when you work with company, then you have other issue in terms of property and management and so on. So I think that this is also another issue that we should really look for the next future more in deep.
Great. I think Annette responded to Theo, and yeah, I think what Theo, if I can paraphrase, is that there's sort of qualities and capabilities and capacities of gene banks that may be beyond the expectations of the ECPGR that are fundamental to define what gene bank operations are. And as we go, so that's the answer to, I think, the question that you posed, Annette, to everyone. I think going back to your first point, though, Annette, you know, I think that if there was a program to address directly the sorts of integration issues, as opposed to the research results, that could be very, very helpful, because those are often being done in small regional networks, but we're reinventing the wheel perhaps in certain areas, and it would be probably very useful. Niels, we are at quarter of the hour. This has been quite engaging. Do you want to continue, or do you want to wrap it up? Or...
Yeah, well, I think we can ask if there are pressing questions to continue now. Otherwise, I think I want to remind everyone that the seminars were recorded and will be available through the website of DivSeek. Yeah. I can only encourage also all partners that have presented today, take the advantage and communicate with DivSeek if you're interested in presenting progress of your projects in a year from now, or whenever you think you have information that is valuable to be disseminated to the DivSeek community. I think, personally, I think it would probably make sense to have a presentation like this perhaps in 2 years from now, that we can present more progress, but probably then we cannot do this anymore in flash talk mode, but we would need more time for individual projects. Or even more preferred, we are back to the time of in-presence meetings, and we can share results and concepts in-person discussions on a conference. So if there is no other question from the audience, if there are questions coming to your mind after the session closes, feel free to send your email to DivSeek. And then I think, Chris, you will have a chance to distribute this to the addressees or to the general audience.
Yep. Send it to me. My contact information is on the webpage. Great. All right. Well, thank you, everyone. I really appreciate your time, and it's very inspiring to see all of these projects progressing. So thank you very much.
Thanks a
Thank
lot.
you. Bye.
Bye.
Great.
Bye-bye.
Bye.
Thanks, Chris.
Thank you.
Everybody.